Okay, so today we are going to see the chapter of biology, biotechnology, principles and processes. Okay, so in order to know this chapter, you need to know little terms, okay, which are there. So first of all, now what is this biotechnology? Okay, so obviously when something comprises of the living organisms which is attached with the technology, we say it's what? Biotechnology. So here, probably what you're doing is you're integrating your basic natural science which is there and all of the technology which can be used at its utmost priority. Okay, those things are called or comes under what? The biotechnology. So related to this, okay, you have certain terms. That is, first we are going to see about the rDNA. Okay, then we have something called as the origin, vector, then plasmid. Okay, then we have nuclease enzymes, palindromic sequence, cloning, and enzymes, that is restriction enzymes. Now these are certain things which are very useful whenever you are doing certain procedures or developing a new product using what? Biotechnology. Now usually this is a combination technology, hence humans are involved, plus the small bacteria, viruses which are there, they are going to play a major role. Because in biotechnology what happens is the bacteria or the viruses which are there, they act as a vehicle. Okay, so when we'll be going forward, you will be knowing that what is a vehicle, and vehicle means usually it's a vector. Okay, so I'll just clear these terms whatever I have written over here. So the first one is the, let's take this rDNA, that is a recombinant DNA. So in the name itself it's there, recombinant DNA means what? Here you're going to use two things. Okay, one is your desired DNA in which you want to make the modification, plus what you're going to use is a vector DNA. So vector means nothing but basically a vehicle which is going to take your DNA into this desired DNA, fuse with the desired DNA, change the characteristic. Okay, and then you are going to what do you say, incorporate this particular modified DNA into your host, so that it will give you the desirable product you're needing. Okay, so this is called as what? Recombinant DNA, that is desired DNA plus the vector DNA. Then you have origin. So origin means usually a particular cell replicates, right? From 2, 4, 8, 16, like that, it replicates. So there is a replication point for every cell or for every DNA. Okay, and with the help of that replication only, we know the mitosis, meiosis, same way it takes place over here. That is, origin of replication means nothing but it's a place which is going to initiate the replication process. Then you have plasmid. Okay, plasmid, and we'll take vector. So plasmid and vector means what? Now plasmid means usually it's a thing, or you can say it's a chromosome which is capable of self-replicating. Okay, so it's a chromosomal DNA usually, but it has a capacity of what? Self-replicating. So if you put something in that plasmid DNA and insert into your host, then what will happen is it will self-replicate. It doesn't need any other source to replicate, and it's going to give you the desired product. So that is called as what? Plasmid. That is it will be mostly like this. Okay, like this it will be. So it's nothing but a circular extra chromosomal DNA which has the capacity of self-replication. Then coming to the vector. So vector means what? It is nothing but a vehicle which usually transfers your DNA or your recombinant DNA to your host. That's called as what? The vector. Next you have is the nucleus. Now these are actually enzymes. Okay, you have basically two types of enzymes. One which is called as an exonucleus and the other one which is called as endonucleus. Okay, exo and endo. So basic meaning you already know, that is exonucleus means what? See, now this nuclease enzyme usually deals with the cutting of the nucleotides. Now you already know what is nucleotides, right? So what does this exonucleus and endonucleus do is, the exonucleus, exo means outer. So whatever nucleotides are present on the ends of the DNA, that it will remove. Whereas endonuclease means what? At a specific position, which is basically called as a palindromic sequence, there it cuts the particular DNA or removes that particular nucleotide from there. Now, what is this palindromic sequence? Okay, so palindromic sequence means, for example, take a particular base pair like ATGC. So this will be having what? It's alternate thing, right? So TACG, correct? So now here palindromic sequence means what? In the same reading frame, okay, you means in this particular frame, you will be having same reading frame sequence, but it will be like a mirror in short. Okay, so actually, if we come to see what is this palindromic sequence means, it's nothing but the what you can say is a same reading frame which is present on the opposite polarity strand of the DNA. Means we know that there are 5 prime and 3 prime, 3 prime and 5 prime, right? So it's opposite whenever you take a DNA strand. So same reading frame whenever you get on the opposite poles, we say it's what? A palindromic sequence. Okay, so this palindromic sequences are very important when you are carrying out a cloning or a replication to produce a particular product. Okay, then cloning. Cloning means what? To obtain multiple copies of that particular rDNA. So it will be like same Xerox copies of that particular original DNA which is there. Okay, so these are certain terms which we are going to see in this chapter in detailed process. Okay, so moving forward, now if we take this biotechnology process, okay, now what actually does it deal with? So you basically have to change a particular DNA structure to obtain your desired product. So obviously it will be needing what? Certain kinds of things. So the first thing is what? A microbe mediated process, in vitro fertilization, synthesis using of gene, preparation of vaccine gene, and correcting of defective gene. Now this is if suppose if you're what do you say, preparing a vaccine. Okay, so when you're preparing a vaccine, you need what? These kind of or these steps are being used whenever you are going to prepare a particular vaccine. Okay, so actually how this is done. Now we already know that recombinant DNA means 2 or more characteristics are incorporated into your plasmid. It is transferred into your host and there it will replicate and give you desired product. So microbe mediated process means what? Here you're going to use microorganisms to transfer that particular what your particular modified gene. Then you have in vitro fertilization. In vitro fertilization means this is done in the laboratories under sterilized condition. Okay, like how in plants you have the greenhouse and then you keep it to the soil so that it can grow and acclimatize to that condition, right? So in the same way you have in vitro fertilization. It's same like that of the test tube babies which we saw in the previous chapters, right? So in this what will happen is you're going to isolate your desired gene and you're going to make out the results in a sterilized condition. So it's called as what? In vitro fertilization. Then synthesize using a gene. Now, already I told you certain enzymes, certain genes you're going to use for a proper process, right? Next is what? Preparation of the vaccine gene. So once you have done the three steps, which is there over here, you're going to prepare that vaccine gene and you're going to check out whether if it's having any defective gene present or not. Means if you have to rerun the process or rerun to make the replicate copies if in case it has a defective gene. Okay, so to do this, there are basic two principles of biotechnology. Okay, that is called as the genetic engineering and bioprocess engineering. So most probably genetic engineering is being used because over here you alter a particular genetic material. Now that can be an RNA or the DNA. And what do you do after alteration of this genetic material? You put it into the host and then whatever the phenotype of that organism is there, that's changed. For example, if you want an insect resistant plant, or instead of a dwarf plant, you want to make it a heighted plant, a tall plant, then what you can do is you can isolate the gene, replicate it, or insert, do certain alteration in your gene. Okay, put that altered gene, that is your recombinant gene, with the help of a plasmid into the dwarf plant. Then what you will see, that that dwarf plant is no more a dwarf, it will become a tall plant. So same way happens even if you're taking a live organism or a plant or microorganism, whatever it is. So that's called as what? Genetic engineering. But bioprocess engineering means what? It is nothing but you are going to basically maintain what do you say, a sterile environment. As I already told you, there should be no source of contamination at all. Okay. And then in an uncontaminated condition, you're going to enable the growth of only those microorganisms which you want. Okay, so only whatever your desired microbe or your desired cell is there, that should grow in large quantities in that what do you say, uncontaminated environment. And when that particular thing grows, you're going to grow it in on a large scale or in a large quantity. For example, if you take industries, there they will grow in a large quantity, right? You have instant yeast also, whereas you have certain yeast which is going to pop up after some hours also, right? So these two are what, like you can say it's a genetic modification which is present in them or which has been done in them. So in the large scale, what you do is you produce whatever your desired things are there, and you make out products such as antibiotics, vaccines, and enzymes. Okay, now you may have seen that there's, whenever you go for a vaccination, okay, or to take certain injections, it will be present in a vial. A glass vial will be there, small. They will break the tip of that vial, insert the injection, and give the injection to you. So that particular tip of that vial for the vaccine, because it has live organism in them, right? So that they are producing that particular vial with the vaccine in a sterilized condition. So this is called as what? Your bioprocess engineering. Okay, so whenever you are going to use any of the principles of biotechnology, now there are certain steps which I have already explained to you, right? So but in short, if you are taking or if you're seeing what is genetic engineering, now this is a major important process which comes into existence when you say the term biotechnology. Okay, so genetically engineered, for example, if you go to your markets, you will see in the vegetables you have hybrid or genetically engineered tomato, okay, genetically engineered what do you say, brinjal, like that many things are there, right? Now sugar cane which is there, every year throughout the year you get sugar cane, right? So what is the reason? They are genetically engineered so that they will grow irresistant of whatever that particular conditions are there, which is just making it too limited what month of growth, right? So if you use the genetic engineering technique, you can just solve these problems and you can make out large quantities but in a healthy variation. Okay, so hence whenever you take a particular DNA, you have to make sure that particular gene from where you're isolating the DNA should be healthy. Okay, then and then only you should insert into the host, replicate it, and introduce into the whatever your desired plant or animal, whatever it is there, right? So this is only the main methods which you're going to use in the genetic engineering. So now coming towards the main part, which is the rDNA or the recombinant DNA. Now already we know what is recombinant DNA, your desired DNA plus your whatever your altered thing is there that you're going to use together, which itself is called as what? A recombinant DNA. So usually to make out a recombinant DNA, you're going to use certain enzymes. Okay, so one such enzyme which you're going to use over here is called as the restriction enzyme. Okay, so when you use this, I'll be telling about the restriction enzyme more. Okay, so in short, if you want to know what is restriction enzyme, it's nothing but a type of nucleus enzyme, which I've already explained, that is the endonuclease and exonuclease. So it's just going to cleave or cut that particular DNA sequences. If it's an exo, that's at the end. If it's an endo, in between only the sequence, it is going to cut your DNA. So that whatever only desired gene you want, that you can take and place it. So in recombinant DNA, restriction enzyme plays a very important role. Then and then only you can produce the DNA, clone it, transfer it, and then you can extract it and make a large scale of it and insert it. Okay, and make what? Desired copies. So how it's actually done. Okay, so now So basically, if you see a particular tool of the recombinant DNA technology, in that the first tool, as I told you, which comes is what? Restriction. And I'll just write it over here. Okay. Because these are important points. So restriction enzyme. So what is a restriction enzyme? It's nothing but a type of nuclease enzyme, which helps in what? Cleavage. At specific sites. So there is that specific site is called as a sequence, right? So we have two types. Already I have told you, exo and endonuclease. Now, for example, the basic or the most common type of restriction nucleus which is being used is the restriction endonuclease called as ECO R1. ECO stands for E. coli and R is the restriction enzyme site which is being used. Okay, so as I told you, whatever the palindromic sequence is there, according to that only you have what? The rest, which site is being restricted or cleaved over there. So E, which is there, it stands for the Escherichia. E. coli's full form is what? Escherichia coli. So it stands for Escherichia. Co is nothing but your species, which is coli. Coli. R is nothing but the strain, whichever you're going to use. And one is the order of isolation. So you will be having echo R1, echo R2, echo R3, so on. Okay. So order of isolation. Okay. And this all deals with your palindromic sequence. For example, if you take a palindromic sequence of the echo R1, so how it comes is you will be having two ends, 5 prime, 3 prime, 3 prime, 5 prime, because it's the opposite polarity thing. And then we have a sequence like this. So if you read from this side or this side, it's the same. Okay, so this has been used what? To cut that particular nuclei and to isolate your desired whatever DNA is there. Okay, so how basically this cutting of the sequence is done? By using your restriction enzyme. Okay, so now over here, if you see, now this is basically the DNA cloning. Before going into cloning, I'll just write the step over here of the restriction enzyme, how it cuts it. Okay, so first is the isolation of DNA. Okay, so whatever desired gene DNA is, that's isolated. Second is what? You have to multiply the copies of that DNA. So it's called as amplification. And this amplification is done by a method called as the PCR. Okay, so PCR is the method which is called as polymerase chain reaction, where instead when you put one, what do you say, a desired gene into that particular machine, it's going to give you hundreds or thousands of copies of the desired sequence gene. That's a reaction, which is polymerase chain reaction. Then what you do is you cleave the DNA. Okay, so both the desired DNA and vector DNA you're going to cleave, but make sure whenever you're doing this cleaving, the DNA which is that is your desired DNA and your vector DNA both should be cleaved by using what? Same restriction enzyme. That's very important. That is same restriction endonuclease. Okay, so once you cleave that particular thing, okay, that is which is shown over here like this. Okay, now if you see vector is there, chromosome is there, you're cleaving it with your restriction endonuclease. Next step is what? The fourth step, ligation. Means joining of your desired DNA with the vector DNA, and finally you insert it into your host. So you get a recombinant DNA with the cleavage of the restriction endonuclease enzyme. So if you see here in this diagram, you have an eukaryotic chromosome. Okay, you have what? A cloning vector, which is nothing but your plasmid, that is extra chromosomal DNA. You just have to cleave it with by using this particular restriction enzyme. Then what you do is you have to insert that rDNA, whatever is there, into your host. Now this is a bacterial cell, so you're just using it into this bacteria, or you're just inserting it into the bacterial cell. So here whatever you see, this is the host DNA, then you have this is the plasmid DNA. After that, what you do is you're cloning. Okay, that is forming multiple copies. And then this process is only called as what? DNA cloning. So whenever you are doing a DNA cloning, basically you use what? A cloning vector. Vector means what? Vehicle. And cloning means exact same copies or the Xerox copies which are there. Okay, so if you see, every sequence is going to have a particular restriction site where this restriction endonuclease will go and cleave it. Okay, so whenever you want to do this process, there is one technique which is called as what? Electrophoresis. What is electrophoresis? Basically, whatever your cleaved DNA sequence is there, okay, you are going to separate them, because sometimes what happens is that DNA will come joined like this. So you cannot obviously use that joint DNA to put it into your desired host. You have to separate each and every sequence, what do you say, clone DNA which is there. So for that, you're going to use this electrophoresis. That is nothing but a process where you have a gel-like cassette. Okay, in that you're going to put a drop of this particular sample, okay, which is containing your desired gene, and you have to run it under by providing the electricity. So once it reaches the end of the cassette, it will be like this. Okay, so you have to pour the sample over here in the slots. Okay, you have to on the electricity, so it's going to run like this. So in whatever slots you have put, it will show you the result, and there will be a selectable marker which is going to indicate that yes, your host or your DNA have been run till here. So it's basically blue color. Okay, transparent gel will be there. In that blue color dots you will be seeing, and that is kept under a scanner, and you will get the result in a what do you say, in a computer. Okay, and then you can see that how many bands are there, okay, how much DNA has been separated and all those things. So whenever you are doing an electrophoresis, it's a simple technique wherein you are going to form certain bands of that particular DNA sequences. Okay. Okay, so now next one is what? The origin of replication. Okay, so now we already know in short what is the origin of the replication, right? So origin of replication means what? It's the specific DNA sequence in the chromosome that's responsible for initiation of the replication. Okay, so for example, if you're having any of the piece of DNA and you just have to multiply it, first you have to know what is the origin of replication. Means what? You have to know where exactly that specific sequence is present, where you can initiate the replication. Okay, so when where in the area from where this replication is going to what you say begin, it's called as the origin of the replication. Okay, then you have the plasmids. So plasmid means, I already told you, small circular DNA which can reproduce on its own. Okay, and they don't have to depend upon the host replicated DNA, because it is having the capacity to self replicate. So hence they are also called as what? Extra chromosomal DNA, and it's a very important part of your genetic engineering if you have to do a gene cloning or a gene therapy. So you usually isolate these plasmids in the bacteria, and few eukaryotes also have this plasmid. And what is the range? As I told you, whatever electrophoresis that we are performing, it separates the DNA fragments on the basis of its size, because the plasmids range starts from 1 to 200 kb. So if you take a small band of that DNA and insert into 200 kb, it's a waste, right? So because it's a see, whenever you're performing genetic engineering, it's very costly. It's not like a few thousands will do it. You need lakhs and sometimes crores to do all that particular processes. Okay, hence whatever according to your plasmid size only, you have to isolate the desired DNA sequence. Okay, so that it can produce whatever your desired enzymes, antibiotics, or whatever are there. So these are certain types of the DNA, like F plasmid, resistant plasmid, viral and degradation, and call plasmids. So depending upon what role it plays, okay, or from where it is originated, these plasmid types are given. Then recombinant DNA technology, I have already told you what it is. Okay, then tools of DNA are DNA technology. So tools means now whenever you're writing about the tools in your exam, you have to mention enzymes. Okay, which are basic enzyme is the restriction enzyme, polymerase enzyme, ligase enzyme. So one will be for the cutting, other one will be for the polymerase means what? To form many copies. Then you have ligase, which is to join again. Then you have vector as well as the host. Then next you have is what? Restriction enzymes. This I have already told you what is a restriction enzyme. Okay, this is what? A palindromic sequence which you can see. Yeah. So if you see here, this ends which are there, it's called as the sticky end. Okay. So sticky end means nothing but there will be a particular replication site, right? So whenever your DNA is originating into two different strands, okay, that end we call it as what? The sticky ends. Okay, so here you can see how that particular sequence is being made and it's been converted into what? Two different strands. So this is the electrophoresis technique which I have just told you. Okay, here one important point in the electrophoresis is the gel which we use. It's called as an agarose gel, which is mostly transparent. Then only you can see how the DNA bands are being separated, and to give that particular color to that particular DNA strands. Because in the OSI, obviously your solution is also transparent, the gel is also transparent. Then how are you going to make out that yes, these are the bands which have been run? So there will be ethidium bromide. Okay, by using the ethidium bromide, which you already put into that gel whenever you're preparing it. So you put that ethylium bromide, and when you observe that particular cassette or that gel under your this one, the computer, you will see the bands because of that ethidium bromide which you have already put. So usually UV lamps which are there, that has been used to expose your bands which is there. Okay. Then the vectors, this is also completed. So two types of mostly the vectors which you use is the bacteriophage and the plasmid vectors. These are the popular vectors which you use to carry your desired gene and insert into your host. Okay, so you can just see what is the difference over here. This is the plasmid, this is the bacteriophage, and this is the cosmids, a combination vector which is there. Then cloning vector, okay, as already told you, cloning vector is nothing but it is just going to make out the clones of your desired DNA, which is a very important tool for the rDNA technology. So whenever you use the cloning vector, okay, there are three things or three properties which you should keep in mind if you're using what? A cloning vector. One is the origin of replication which is there. Second is whatever marker you are going to use, a selectable marker is being used over here. And third you're going to use over here is what? The cloning sites. Only if these properties are feasible for that particular cloning vector, then only you can use it, okay, as a cloning vector. Okay, so